Good morning students. So in the last class we have discussed about fermentation process. In the fermentation process we have discussed about upstream processing, right? So now in continuation to upstream processing, that is in the fermentation or in any fermenter, after the fermentation process it is called as downstream processing. Okay, so now in the downstream processing it may be either in situ or you can get an intracellular product or extracellular product. So after fermentation process you are going to get all the products. So now you are going to harvest the cell. Okay, that is you are going to separate the solid and the liquid media, or you are going to separate or harvest the cell. From the harvested cell, if it is an intracellular product, okay, if the product is intracellular, you can able to get, or either you have to do again cell destruction process. That is to break the cell and you have to get. So this can be done either by means of centrifugation or filtration. Centrifugation or filtration. Okay, so first after the fermentation process you are doing, or you are harvesting the cell, that is you are separating the solid and liquid media, and once the cells are done, if the product is intracellular, that is the product is present inside the cell, that is called as intracellular product. So that intracellular product needs to be lysed because the product is present within the cell. So the cell has to be lysed, that is the cell wall has to be cleaved, and then you have to get your product which is present inside the microorganism. That can be obtained by means of cell disruption. So cell disruption can be achieved by means of centrifugation or by means of ultrafiltration process. Then you get a cell-free extract, that is only product or the crude product. This is called as cell-free product. So till this stage is called as primary recovery. Okay, so first you are harvesting the cell, then if the cell is intracellular, then you are going for cell destruction, that is you are done by means of using centrifugation or ultrafiltration process. So then finally you get cell-free extract. So this stages is all together called as primary recovery, that is you are getting your product, that is ethanol which is present inside the microorganism. You are remove the microorganism and now the ethanol is present inside the microorganism. So now you are lysing the microorganism and releasing the ethanol outside the microorganism. So then you are centrifuging or filtration, and finally now you are removing the microorganism and only ethanol or crude ethanol is present. That you need to do. So this can be achieved by means of dialysis, okay, and precipitation, partition chromatography, partition chromatography, and obviously like ultrafiltration, distillation. So now we are talking about ethanol. So distillation is also an important step in this process. This is called as product purification step. Okay, so this stage is called product purification, and finally the next stage is like crystallization, crystallization, drying, lyophilization is all the process is called as finishing process, that is it is making into a final product, and finally you are doing and packaging, that is you are getting your final product. This is only for intracellular product, that is you are harvesting the cell, and if the product is intracellular, you are lysing the cell wall and getting the product, that by means of centrifugation or ultrafiltration process, and you are separating the cell and your crude product. This all together called as primary recovery process. Once your primary product or your product is obtained which is in a raw form that you need to purify. Say for example, whenever we state that 100% sugar, okay, so when we talk about sugar, that is obtained from sugarcane juice. So that is crushed and juice is obtained and which is boiled, so you get a sugar, okay. So sugar molecules is obtained. Say for example, we get some sugars. If that's a condition, say for example, when a person comes to your home, a guest, you want to prepare a tea, okay. If the sugar molecule is not in a shape, you don't know how much of sugar that you want to put. One is like 10 gram, one is in a 5 gram, and one is, we don't know the weight. Then exact weight or composition, you don't know. So in order to give one proper appearance and to measure easily by the consumer, you are making it as a final product in a particular shape, size, color, etc. So now, initially you have recovered the product, but the product will not be 100% ethanol. So the maximum the microorganism could be able to produce only 15% concentrated ethanol. Okay, then in your lab you are using 100% concentrated ethanol. How do we get right from 15% to 100%? So you have to purify and to get a final product. So here that's what product purified state. So in this state, either by means of dialysis, precipitation, partition chromatography, distillation, in any one of the process, you can purify the raw product what you have obtained. So here ethanol. So ethanol can be done by means of precipitation or distillation. Most probably 99.99% will go with distillation process. So distillation process by means of heating, or you are separating the compound by means of its boiling point. So by means of distillation, you are increasing the concentration of ethanol which is obtained as a primary product 15% to 100%. Okay, this is called as product purification process. Finally, from 99 or 100%, either by means of drying, lyophilization, crystallization, or any fraction instead of distillation, advanced distillation process, you are obtaining 100% absolute concentrated ethanol. Okay, this is called as finishing process. Once the process is finished, you are going for packaging. So this is absolutely called downstream processing. So whenever or whatever the product is obtained, in each stage it will be treated as effluent. Okay, the product, that is once you are harvesting, the liquid will come out. Okay, then when you are doing cell destruction, you are removing the microbial cell. What? Then dialysis, the unwanted compounds which is present in the ethanol will come out. So all are altogether called as effluents. So this is for intracellular compound. The same thing for extracellular, that is the product which is released outside will be taken into the spent medium. Okay, from the spent medium it will be considered as extracellular. If the product is extracellular, then we have to go for concentration of product. So then again you come for product purification, because here in the concentration step, that is the product which is already released into the medium. So you are just removing the microorganism from it, because already the medium consists of the product. So now you just need to remove the microorganism alone, and the media which is liquid portion, you need to go for concentration process so that you get your product. So that product again will go for product purification process and finished process. This is altogether called finished processing or downstream processing. Okay, so this upstream and downstream together come into view of fermentation technology, and all this process has an unit. All this process is altogether called as unit operations. So each and every unit operation plays a very vital role. Sometimes the combination of unit operations or combination of these techniques are employed to get a final product. Okay, so this can be further explained in many ways how to get. say for example in a cell disruption process, how to disrupt the cell. So disruption can be done either by means of physical, chemical or by means of biological process. So there are variety of techniques and unit operations are available to get your products. Okay, so this is the basics of upstream and downstream processing. Okay.